Hello everybody, it is Mr. Moreau again with a quick introduction to our Cytochrome C comparison lab. Now what this lab is going to help us understand better is one of the components of the evidence that scientists use to support the theory of evolution. We know that it has happened, we know that it will continue to happen, but what scientific information can we study that helps us understand better how scientists can in fact confirm that evolution has, is, and will continue to occur? One of those major sources of info is biochemical comparisons. By biochemical, I mean molecular comparisons. In this case, like RNA, DNA, amino acids, proteins. Well, the protein that we are going to be studying in this specific example is cytochrome C. By comparing the cytochrome C protein across many different species, we are kind of indirectly comparing the DNA across different species, because we know that the protein of cytochrome C comes from an amino acid chain, which is coded for by the DNA, and what we learned about protein synthesis teaches us the connection between molecules like RNA and DNA to the proteins that we are studying here. So the quick intro of the methods we will use for this lab are seen on page 1. Now these are the techniques that we are going to be seeing, or actually putting into use on pages 2 and 3. So you can see that we are going to be given multiple rows of amino acids of different organisms. Okay, so every different amino acid has been assigned an alphabetical letter in this example, so we don't have to write out the three-letter beginning for each amino acid, or we don't have to write out the name of every amino acid. Every amino acid has just been assigned a single letter, and that's what all these letters are here. So the top row in this little tiny example shows us the amino acid sequence for cytochrome C of one specific organism, and then the row right below it, that shows us the amino acid sequence for cytochrome C of a different organism. And you can see that they're not exactly the same. In fact, in green, 9 different differences have been highlighted for you. So that's kind of the method that we're going to be doing. We're going to be provided different rows for different organisms of what their amino acid sequence is for cytochrome C, compare them to each other to determine evolutionary relationships between them. Eventually, you're going to count those differences. So in what we see in method A1, those two rows, I count 9 different differences. Perhaps there'll be more or less in different comparisons that you make throughout this lab, and you'll record that number like you see here in the correct space where it tells you which organisms you're going to be comparing. Okay, we're not going to be sharing data with the rest of class. I'm simply going to split this activity up into 2 smaller segments so that we have no problem working on our own here while we are absent from each other. Now what we're eventually going to do is use those number of differences to fill out a phylogenetic tree or a branching tree or a cladogram, and we're going to follow the directions here to kind of help us figure out how that works. All right, and here's the bulk of the info. So I'm going to zoom out here so I can see the entire page. So what this shows us is several different organisms. You can see those written in the far left column there. You can see that human and chimpanzee share a row because they have identical versions of cytochrome C. The second row is the rhesus monkey. The third row is the horse. The fourth row is the donkey. The fifth row is the common zebra. The fifth row is the pig, cow, and sheep. They share a row again because the pig, cow, and sheep version of cytochrome C are identical to each other, so we can simply put them all on the same row. Now what I've done for you as an example is I've highlighted the row for the common zebra and the chicken/turkey. Okay, so chickens and turkeys have the same cytochrome C version, but it is different than the zebra version. Now what we can see here is that the number of amino acids goes all the way from number 1 up to 112. I know that because up in the top row, it shows me, I'll go to a different color here, the amino acid number starts out counting from 1 up through 10, 20, 30, 40, 50, 60, all the way up to 112. It continues on in the second row. They just didn't have enough room on one single page. Okay, so what I have in yellow, I've highlighted the entire sequence for the common zebra, and I've also highlighted the entire sequence for the chicken/turkey. And notice that I have highlighted in green wherever I see a difference. Right there, that is a different amino acid for the chicken turkey than it is for the common zebra. Okay, so I've highlighted that in green, and you can see that I've done that a few times throughout these rows. So if I was required to figure out the number of differences I see in the cytochrome C protein between the common zebra version versus the chicken/turkey version, I would simply count up those differences, and that's what I'm going to fill out in this next table. So what I showed you above was comparing the zebra to the chicken/turkey. Now I wanted to count one out for you that was not part of your table, so I added the zebra as an extra column off there to the right. And when I went through those rows and counted the differences, I counted a total of 9 differences. So that's what I put. That's why I put number 9 in the box where chicken and zebra meet in this table. You don't need to worry about these. These are shaded out because they would just be repeats, right? For example, the yeast and snake box, you can see I've highlighted in green. Why would I do it, twice if I'm going to this one here? Okay, that is also where yeast and snake meet, so that's why they've shaded everything out on one side there. Well, that's the approach you will take after figuring out how many differences there are between the cytochrome C protein of these various organisms. You're going to do your best to try to figure out where you would put them on this cladogram or branching tree. Like the directions above say, whichever ones have the fewest differences, that seems like it would suggest that they have the most recent common ancestor. These two organisms here share the most recent common ancestor. So this organism and this organism would be the two out of what you see in table 1 that are the most similar to each other. Okay, and that's where we'll stop for today. I'm going to have you, you feel free to work ahead and finish the questions if you like, but we're going to give you the opportunity to save that for the time we spend on this on Monday. Okay, so I expect you to get through page 3 for our e-learning day today. And that'll do it. All right, folks, see you later.